السلام عليكم أعزائي طلبة السادس العلمي لمدارس المتميزين وكلية بغداد. نبتدئ درسنا السادس لهذا اليوم من مادة الأحياء إن شاء الله بموضوع Cell Activities الأنشطة الخلوية. In today's lecture, we will start with cell activities. What are cell activities? Cells, whether they were plant or animal, achieve many activities. These activities can be summarized as follows. الخلايا سواء كانت نباتية أم حيوانية فإنها تنجز بعض الأنشطة المختلفة، وهذه الأنشطة يمكن إيجازها فيما يلي. What are these cell activities that we are talking about? The first activity is the exchange of materials through membranes, while the second activity is cell metabolism. النشاط الأول الذي تقوم به الخلايا هو تبادل المواد عبر الأغشية أو عبور المواد عبر الأغشية. أما الثاني فهو الأيض الخلوي. If the question was fill in the blanks with the missing words, and he was asking about enumerating the cell activities, and we have two blanks in the question. So the first blank will be filled with the word with the exchange of materials through membranes, and the second blank answer would be cell metabolism. So this is very important question, and sometimes students don't realize that, so they suppose that the question was about diffusion or permeability or so on, but really the question is here about the cell activities. When it asks about it, the answer would be only two. The first one is the exchange of materials through membranes, and the second is cell metabolism. إذن عندما يسأل بالامتحان عن ما هي الأنشطة الخلوية التي تقوم بها خلايا الكائنات الحية، وممكن يكون السؤال بهيئة فراغات، فيكون هناك فراغين لتكملة الجملة، فيكون الفراغ الأول يملأ الإجابة الأولى التي هي تبادل المواد أو عبور المواد عبر الأغشية، والفراغ الثاني سيكون الأيض الخلوي. فكيف تعبر أو تمر المواد عبر الأغشية? How does that happen? How can exchange of materials through membranes occur? Exchanging of materials inside and outside the cell is considered the basic cell operation. This operation regulates cellular functions and determines exit of waste materials and water outside the cell. It is essential, the exchanging and the passing of materials is essential. Why? It is essential to maintain biological operations of the cell, and second, construction of living matter. تعد عملية عبور المواد من عبور المواد إلى الخلية وخارجها من العمليات الخلوية الأساسية. هذه العملية، عملية عبور المواد، تنظم الأحوال الوظيفية للخلية. التنظيم هذا عندما يحصل بالنسبة للخلية ماذا يؤدي؟ من خلاله يتم تحديد، بموجبها يتم تحديد وتنظيم خروج المواد الإخراجية والماء من الخلية، ولا يخفى من أهمية لذلك. أهميته في أين؟ في حفظ maintain، في حفظ واستمرار العمليات الحيوية للخلية، وثانيا في بناء المواد الحية فيها. إذن هنا يعتبر هذا سؤال مهم، وقد يكون السؤال بهيئة تعليل explain exchanging of material inside and outside the cell is considered the basic cell operation. The answer would be this operation regulates cellular functions and determines exit of waste materials and water outside the cell. It is essential to maintain biological operations of the cell and construction of living matter. Also, the question may be the process of exchanging materials through membranes is considered one of the basic cellular process that regulates cellular functional conditions. The answer for this question will be it is essential to first maintain biological operations of the cell, and the second is it is used for construction of living matter. إذن عندما يسأل عن أهمية عبور المواد عبر الأغشية وكيف لها دور في تنظيم خروج المواد الإخراجية والماء من الخلية، وما أهمية ذلك، ما أهمية خروج المواد الإخراجية والتبادلات الحاصلة عبر الأغشية، فتكون الإجابة بأولا أنها سوف تحفظ وتساعد على حفظ واستمرار العمليات الحيوية للخلية، وثانيا فإنها تقوم ببناء المواد الحية فيها. فممكن يكون السؤال بهيئة تعليل، أو ممكن أن يكون بهيئة أنه ما أهمية هاي العملية اللي هي تبادل المواد أو عبورها عبر الأغشية. The passage of materials is done via many ways. When we said the first activity was the exchange of materials or the passage of materials through membranes, the passage can be done by 7 different ways. What are these ways? The first way is diffusion. The second way is permeability. The third is osmosis. The fourth is active transport. The fifth is phagocytosis. The sixth is pinocytosis, and the exocytosis, which is the last one. These 7 ways are examples for the exchange of materials through membranes. إذن عبور المواد عبر الأغشية تتم بعدة طرق، وهي 7. ما هي هذه الطرق؟ الأولى هي الانتشار diffusion، الثانية هي النفوذية permeability، الثالثة هي التناضح osmosis، الرابعة هي النقل النشط أو الفعال active transport، الخامسة هي phagocytosis الأكل الخلوي أو البلعمة، والسادسة pinocytosis أو الشرب الخلوي، وأخيرا الإخراج الخلوي exocytosis. إذن 7 طرق تستخدمها الخلايا لأجل عبور أو مرور أو تبادل المواد عبر الأغشية. The first way is diffusion. So we're going to take cell activities. These cell activities, the first one is exchange of materials through membranes. The second one is cell metabolism. When we come to the exchange of materials, it is done in 7 different ways. The first way is diffusion. What is diffusion? Diffusion is defined as movement of ions and particles in a certain medium from high concentration regions to low concentration regions. الانتشار هو حركة الأيونات والجزيئات خلال وسط معين من أماكن ذات التركيز العالي إلى أماكن ذات التركيز الواطئ. هذا هو الانتشار. ملاحظة مهمة جدا، الانتشار قد يحصل عبر الأغشية وقد يحصل لكن ليس عبر غشاء، وإنما خلال فد ميديم معين، خلال فد وسط كالهواء كالماء. هذه تعتبر أوساط للنقل. فإذا الانتشار ممكن يحصل عبر الأغشية أو خلال أوساط لكن دون وجود غشاء. As an important note, diffusion, we usually know that diffusion occurs through certain mediums, or sometimes it can occur through membranes. So diffusion does not occur only through certain mediums. Certain medium may be a gas medium or a liquid medium, sometimes solid medium, but sometimes it can occur through a membrane also. And now we are going to take in some examples for which types of particles or ions or molecules that can go through diffusion that occurs to them and how. As a rule, gases like O2 and carbon dioxide, these are two types of gases that can diffusion occur with them. The second example are materials. The first example is the gases. The second example is materials. The materials that are soluble in fats. Examples for materials that are soluble in fats are hydrocarbons and alcohols. So diffusion, when we need an example for materials or subjects that can go through, diffusion occurs with them. The first one is gases. Gases like O2 and carbon dioxide. The second example is for materials. Here is the soluble materials that are soluble in fats. Example for these materials that are soluble in fats are hydrocarbons, and the second example is alcohols. If I ask about why, especially materials that are soluble in fats, why? The answer would be, if we are talking about the plasma membrane, the cell membrane, which is known as plasma membrane, and we know that the plasma membrane is composed of two phospholipid, as we said, layers. So we said phospholipid, that means lipids. And there are proteins also, but because they have these phospholipids, so the materials that are soluble in fat can easily go and pass through the cell membrane. So that's why he said here materials especially that are soluble in fats. So the gases, for example, O2 and CO2, and the second example, which is materials that are soluble in fats like hydrocarbons and alcohols. إذن المواد التي لها القابلية على المرور عبر الأغشية أو يحصل فيها ديفيوجن بشكل واضح هي أولا الغازات، ومثال عليها غاز الأكسجين وعندنا ثنائي أكسيد الكربون. وثانيا المواد materials اللي تكون من ضمنها المواد التي لها القابلية على الذوبان في الدهون مثل الهيدروكربونات والكحول. فهذه المواد, these two subjects, the gases and these materials, are both of them can spread through biomembranes. That means the cell membranes freely. These gases and this type of materials can spread through biomembranes freely. Also, this is an important note. Maybe the question will be list the materials or the subjects that can or particles that can pass or spread through biomembranes freely, and the answer would be the gases and materials with the examples. Here we are going to take an experiment about diffusion. This can be observed. How can we see the diffusion? This can be observed by the naked eye when copper sulfides or potassium permanganates are placed in water. The colored material spread through the water when crystals of above materials dissolve in water. When can these colored materials spread through water? The answer is when crystals of these materials, copper sulfides or potassium permanganates, dissolve in water. In this experiment, we're going to take solid particles which are copper sulfides. Copper sulfides are usually, they have blue color, while potassium permanganates, they have purple in color. They are usually purple. When we place one of these solid materials inside water, the water will be colored by these materials. For copper sulfide, which is blue, the water will become blue in color. But if we use potassium permanganate, the water will be purple in color. So the material of the copper sulfide or potassium permanganate will spread through the water. When that happens, it happens when the crystals of these materials dissolve in water. So the colored material spread on a short distance, and it hardly spread for a long distance. عندما نأخذ هذه المواد الصلبة، مثلا لدينا باللغات كبريتات النحاس التي تكون زرقاء في لونها، أو نأخذ بلورات برمنجانات البوتاسيوم وتكون لونها بنفسجي، ونضع إحداها في داخل إناء زجاجي يحتوي على الماء. سوف نلاحظ انتشار سبرد حيحصل، نلاحظ انتشار المادة الملونة. هذه المادة التي تنتشر والملونة كيف نراها أو متى؟ نراها عندما يحصل ذوبان. ستذوب البلورات لأي من هذه المواد علاه تذوب في الماء، فعندما تذوب سنراها تنتشر في داخل الماء. هذا الانتشار سيحصل عبر المسافات القصيرة، لكن يقل انتشارها عبر المسافات الطويلة. لماذا يكون انتشارها في المسافات القصيرة أسرع وأعلى مما يحصل انتشارها في المسافات الطويلة؟ السبب في ذلك هو the distance of spreading particles is directly proportional to square root of time for spread. أي أن انتشار المواد عبر المسافات القصيرة يقل، انتشار المواد عبر المسافات القصيرة يكون أعلى بينما يقل عبر المسافات الطويلة. هذا يعود إلى المسافة التي تقطعها الجزيئات المنتشرة. هذه المسافة التي تقطعها الجزيئات المنتشرة تتناسب طرديا مع زمن الانتشار. كلما كانت المسافة التي تقطعها هذه المسافة هنا، هذا إناء زجاجي وهذا إناء زجاجي آخر أكبر منه ويحتوي أيضا على ماء، فالمسافة هنا التي تقطعها الجزيئات في انتشار هذه المسافة قليلة، فتحتاج إلى زمن أيضا قليل للانتشار. أما عندما تكون المسافة كبيرة فأيضا الجزيئات تحتاج إلى زمن أطول للانتشار، لذلك تكون العلاقة هنا علاقة طردية بين المسافة التي تقطعها الجزيئات مع زمن الانتشار. هذه علاقة طردية، كلما كانت المسافة قليلة كان الزمن المحتاج أيضا قليل، وكلما كانت المسافة طويلة نحتاج إلى زمن أطول لانتشار المادة في الماء. What really happens here, now we have this it may be a glass basin, or it may be a glass of water or a beaker, but it should be from glass so we can see what happens inside it. And inside this glass of water we can see the particles of water here as in the book. Water molecules are known as solvents because the dye molecules are known as solute. They will dissolve in the solvent. The solute dissolves in the solvent. لدينا هنا جزيئات الماء التي سوف نقوم بتلوينها باللون الأزرق هذا بشكل واضح، التي تعتبر المذيب. ولدينا جزيئات الصبغة هنا في الأسفل التي هي المذاب solute، وهذه جزيئات الماء. When we put the potassium permanganate or copper sulfide, it will go first to the bottom of the glass. Afterwards it will start to spread. عندما نقوم بوضع جزيئات وبلورات كبريتات النحاس الزرقاء أو بلورات برمنجانات البوتاسيوم البنفسجية، في البدء تنزل إلى الأسفل، فهي ثقيلة، تنزل إلى الأسفل الإناء، ثم تبدأ عملية الانتشار بصورة واضحة. Here we have a high concentration of the solute, while here we have high concentration of the solvent. This solute here is going to change its place and move from its high concentration to low concentration of the solute. Here we cannot find any particles of the solute, while here we have high concentration of the solute. So it will move from the high concentration of it to the low concentration for it. هنا جزيئات المادة المذابة توجد بتركيز عالي، بينما هنا لا يوجد جزيئات لهذه المادة، أي نعتبرها تركيز منخفض للمادة، فتنتقل هذه الجزيئات من أماكن ذات التركيز العالي لها إلى أماكن ذات التركيز الواطئ لها. كذلك بالنسبة لجزيئات الماء، هنا ذات تركيز عالي وهنا تركيز واطئ بالماء، فتنتقل جزيئات الماء من أماكن ذات التركيز العالي لها إلى أماكن ذات التركيز الواطئ لها. So also here for the solvent, which is the water particles, here we have high concentration of water particles, while here low concentration of water particles. So they will move from high concentration to low concentration of water. When the movement here for these particles occur, we say the process here is the diffusion that has happened. So here is the water molecules, which is the solvent, and And here is the dye that we are using, dye molecules, which is the solid. Afterwards, what happens in a period of time? These dye particles are going to move from its high concentration here to its low concentration there. And this is the arrow that shows how the dye is going to move. The arrows to show the direction of movement for the particles. And for water particles here, the solvent, we are going to also draw blue arrows to show the direction for water particles. Direction of water particles from high concentration of water to low concentration of water. And here, water particles between dye particles. Here, what occurred is the spread of particles these particles are spread of dye particles in water. In the last step here, after a period of time, we will see that there is equal of spread for particles of water and the dye. So all the glass will have the same color. Here, maybe more purple here, and if it was potassium permanganate, but afterwards all the glass will have only one color. All of it will be purple or blue depending on the type of dye that we have used. So here it will be equal spread for the dye particles and water particles. So maybe in the exam it will ask about, wants you to illustrate how diffusion experiment occur. Maybe the question will be explained by an experiment the diffusion of materials in a certain media. So the student should write all the stages that happened here, which is when we are using copper sulfide or potassium permanganates, when they are placed in water. So the colored material spreads through the water when crystals of these above materials dissolve in water. So with the passage of time, matter will spread all over the water as colored materials spread on a short distance, and it hardly spread for a long distance. So this will be the answer, the appropriate answer for this question. And another question may be, why does colored material spread on a short distance and it's hardly spread for a long distance? The answer would be because of the distance of spreading particles is directly proportional to square root of time for spread. So this was the diffusion process. When the question in the exam was about the diffusion process, even if it never asks about the drawing here for this experiment, the student should draw it even if he wasn't asked. Even if he wasn't asked to draw this process, the student should draw it without any question about it, because you cannot say something about an experiment without doing the drawing of it to show what really happened. So the drawing here should be accompanied with the answer about talking about the stages of the experiment and what happens during this process. إذا بعدما يتم الطلب من الطالب بشرح عملية الانتشار، حتى لو لم يتم الطلب من الطالب الرسم، فيجب عليه أن يقوم برسم هذه العملية، عملية الانتشار، وتوضيح الخطوات حتى لو لم يطلب منه في السؤال، لأنه لا يكون هناك تجربة بدون رسم. الرسم عادة ودوما يوضح ما حصل من أحداث تم شرحها كإجابة للسؤال في الامتحان. إذن لاحظنا هنا كيف أن جزيئات الصبغة انتشرت ضمن جزيئات الماء، وعادة هنا الانتشار عندما يحصل هو وجود فراغات ما بين جزيئات الماء. هذه الفراغات تساعد على انتقال جزيئات الصبغة بين جزيئات الماء، وكذلك أيضا جزيئات الصبغة بينها أكو فراغات، فتنتقل جزيئات الماء عبر الفراغات الموجودة بين جزيئات الصبغة. So here when we realize that there are spaces between the particles of water, also there are spaces between the particles of the dye. So the water particles, how can they diffuse in here, and the dye particles, that is the water particles pass through these spaces that are between the dye particles. Also the dye particles enter between the spaces here, the spaces between the water particles, so this will make the diffusion process easily. Second way for exchange of materials is diffusion. It's permeability. The first one is diffusion and the second way is permeability. النفوذية. The second one is permeability. Permeability, it is exchange of materials between the cell and its environment by aid of plasma membrane. إذن بالنسبة للنفوذية هي ظاهرة تبادل المواد بين من؟ بين الخلية ومحيطها عبر الغشاء البلازمي. إذن يؤكد هنا أن النفوذية، عندما نقول النفذ، عادة النفوذية تحصل عبر الأغشية، بينما الانتشار، عندما ذكر الانتشار، ممكن يحصل خلال الوسط، كما هنا وسط مائي ليس غشاء. عندما حصل الانتشار خلال وسط، وممكن أيضا خلال غشاء، لكن النفوذية عادة تحصل عبر الأغشية. So here in permeability we realize that it mentioned here, it occurs by the aid of plasma membrane, while in diffusion we realized that diffusion can occur through a central medium or through plasma membrane also, while permeability is through plasma membrane only. And I have another note here that when he said about the gases here, gases, when sometimes we are inside a room, I'm sorry because I forgot to mention it, when we are inside a room and here is the door, and we enter here from the door and we have a perfume in our hands. When we spray this perfume here in this corner, after meanwhile we will realize that the perfume will spread in all of this room. So how did the particles of this perfume pass here through all the room? It's the same way what happened here between the dye and the water particles, because the particles of the perfume, they found here some spaces in the gas here of this room. So we know that there are lots of gases usually in everywhere in the room and outside it. So these gases are composed of particles. So the perfume particles pass through the spaces that are found between the gas particles inside this room and reach every corner of it. And this way we find that diffusion here occurred through a certain medium, and this medium is a gas medium, while here the medium was a liquid medium, which was the water. إذن أيضا ما يخص الانتشار، كما ذكرنا انتشار المادة الصلبة في داخل الماء هنا بعد أن ذابت، أيضا هناك انتشار للغازات عبر وسط معين، والوسط هنا ليس سائل كما حصل هنا. هنا كان الوسط سائل الماء، أما هنا الوسط هو غازي. نعلم أن الغرفة مليئة بالهواء الجوي وهو كله غازات، مجموعة من الغازات، وجزيئات الغاز هذه أيضا توجد بينها مسافات. فجزيئات العطر انتشرت بين الفراغات، هذه السبيسز الموجودة ما بين جزيئات الغازات الموجودة ضمن هواء الغرفة، فانتقلت جزيئات العطر من هذه الزاوية إلى جميع الزوايا. فبعدها نجد بعد فترة معينة نجد أن الغرفة مليئة بهذا العطر الذي تم رشه في زاوية معينة. أيضا هنا ما حصل هو أيضا diffusion process، أيضا هي عملية انتشار، لكن انتشار جزيئات الغاز ضمن أيضا جزيئات غاز أخرى. أما بما يخص النفوذية permeability، كما ذكرنا هي أن يحصل هنا تبادل لمواد ما بين الخلية ومحيطها، لكن أكدنا أنه يحصل عبر الأغشية كما في الغشاء البلازمي plasma membrane. The cell can absorb food materials in an appropriate food medium. This does not mean that food outside the cell can all be absorbed by the cell. أي يقصد هنا أنه تستطيع الخلية أن تمتص المواد الغذائية إذا وجدت في وسط غذائي مناسب، لكن هذا وجود هذه المواد الغذائية خارج الخلية لا يعني بالضرورة أنه بإمكان الخلية أنه تمتصها وتستغل هذه المواد الغذائية. إذن هذا يعني إذا كان هناك خلية معينة ضمن وسط، كلما وفرنا غذاء، هل هذا يعني أنه يجب على الخلية امتصاصها؟ لا، الخلية تمتص احتياجها فقط، الفائض لا تقوم بامتصاصه، إذن تمتص حسب حاجتها، عادة هكذا يحصل. زين، إضافة إلى هناك شروط لمرور هذه المواد عبر الأغشية، شروط بالمادة كيف يسمح لها بالمرور عبر الغشاء. فالمرور ليس كما تشاء المادة، وإنما كما يشاء الغشاء البلازمي. So here, as it says, this does not mean that food outside the cell can all be absorbed by the cell. But here are some features which are very important for the materials, so they can be allowed to enter or pass through this plasma membrane by permeability. The first one is these materials must pass through the plasma membrane. It means that they have the ability to pass. And the second is these food materials should have certain solubility in water to pass through this membrane. Similarly, excess materials and feces should dissolve in cytoplasm to pass through the membrane out of the cell. إذن نحن قلنا إنه ليس كل ما موجود يجب أن تستغله الخلية، لكن هناك شروط. فهذه المواد، على هذه المواد أولا أن يكون لها القابلية أن تمر عبر الغشاء البلازمي. كما هذه المواد يجب أن تتميز بأن لها القدرة على الذوبان في السيتوبلازم. طبعا بالنسبة للمواد التي تدخل إلى الخلية يجب أن يكون لها القدرة على الذوبان في الماء حتى تمر. أما التي في داخل الخلية وتريد أن تخرج إلى الخارج يجب أن يكون لها القابلية على الذوبان في السيتوبلازم. إذن هذه المواد يجب أن يكون لها القابلية على أن تمر خلال الغشاء البلازمي. فإذا كانت تريد أن تدخل من خارج الخلية إلى الداخل فيجب أن يكون لها قابلية على الذوبان في الماء. وإذا كانت هي مواد اخراجية أو فضلات وتريد أن تخرج من داخل الخلية إلى الخارج عبر الغشاء البلازمي فيجب أن يكون لها القابلية على الذوبان في سيتوبلازم الخلية حتى تستطيع أن تمر عبر الغشاء البلازمي وتخرج خارج الخلية. So here we can find that there are some factors that determine the degree of permeability. First one is these materials must pass through plasma membrane. The second factor is food. These food materials should have certain solubility in water to pass through this membrane, and similarly the excess materials and feces should dissolve in cytoplasm to pass through the membrane out of the cell. This question is very important and its answer, and I'm going to repeat the question again, it is what factors determine the degree of permeability. Here also we have a note here. The permeability is the exchange of materials between the cell and its environment. All food materials should what? Should have certain solubility in water to pass through this membrane, as I said here. This is when the materials need to enter the cell, while the materials that want to access or want to exit the cell, they should have the ability to dissolve in cytoplasm. Also here it says generally membranes are classified according to permeability into. The question here would be classify membranes according to permeability. Here the question was asking about the features for the material that can pass, but here the question is about the membranes, because there are different types of membranes. So membranes are classified according to permeability. إذن هنا عندما يكون السؤال صنف الأغشية تبعا لقدرتها على نفاذية المواد، فتقسم الأغشية إلى the first type of membrane is permeable membranes, permeable membranes. The second type is semi-permeable membranes. The third type is selectively permeable membranes. And the last one is non-permeable membranes. We come to the first one. Permeable membranes. These membranes allow passage of materials regardless of their structure and size. Here he said it allows. It didn't say here it controls, because this type of membrane does not control, it only allows. That means it's all the time it's open. It allows any type of material to pass through without any notes, without any blocking from it. So that's why it allows for these materials regardless of their structure and size. So it does not give attention to the size or the structure of this material. So it allows all the time for all types of structures and structures for materials and all sizes of materials to pass through it. An example for this type of material, we have the cell wall. Cell wall for plants, as we know, are permeable membranes. النوع الأول من الأغشية هي الأغشية ذات النفاذية التامة، هي البرميبل ذات النفاذية التامة. هذه الأغشية تنفذ المواد من خلالها بغض النظر عن طبيعتها أو حجمها. لا تهتم إلى طبيعة أو حجم الجزيئات المارة من خلالها. ومثال على ذلك هو الجدار الخلوي. So these membranes, as I said, they allow. هذه الأغشية تسمح ولا تسيطر ولا تتحكم. سابقا عندما أخذنا الغشاء البلازمي ذكرنا أنه يتحكم، أما هنا يسمح. Allow, that's all. The second type of membrane is the semi-permeable membranes. These membranes do not allow equal passage of solutes as level of solvents. These types of membranes, they control. They do not allow only, they control what passes. So what passes through the membrane are different types of particles. Some of these particles are solutes and some of these particles are solvents. So it does not allow an equal passage to solvents and solutes at the same time. Sometimes it allows for solutes more than solvents, and sometimes it allows for the solvents more than the solutes. So it does not allow equal passage of solutes as the same level of the solvents. And here we really have an example for it is the plasma membrane. The plasma membrane. It says it is semi-permeable. Where is it mentioned like that? Afterwards, when we are going to take the osmosis here, we're going to see it says a semi-permeable membrane as plasma membrane. So a semi-permeable membrane is a plasma membrane. So it controls what passes through it. إذن الأغشية اللي هي semi-permeable هي الأغشية ذات نقول عنها شبه نفاذة. فهي لا تسمح بعبور المواد الذائبة بنفس معدل عبور المواد المذيبة، فهي تسيطر على ما يمر من خلالها. والمثال عليها هو الغشاء البلازمي، ونأخذه فيما بعد في موضوع التناضح الأوزموزية. يذكر هنا أنه الغشاء شبه النفاذ مثال عليه هو الغشاء البلازمي. The third type of membrane is the selectively permeable membranes. Selectively permeable membranes, these membranes allow selective passage of materials according to size of the particles. These type of membranes also controls, so they select what material is allowed to pass according to its size. That means it never lets the passage of materials as they want only. No, it enables the passage of particles through some important conditions. One of these conditions is depending on the size of this particle. The example here for selectively permeable is also the plasma membrane. A student could ask, what is the difference between selectively permeable and semi-permeable? And is it correct that plasma membrane is selective permeable and at the same time it's semi-permeable? The answer would be plasma membrane is selectively permeable, yes, because it allows some particles to pass while it doesn't allow for others to pass alike. Like an example that means is protein particles. It doesn't enable protein particles to pass through permeability. Protein particles are considered huge in size, particles that are large in size. They are not small. So they cannot pass through plasma membrane easily. And they cannot pass through plasma membrane by permeability. They need another process, another way to enter to the cell. So the plasma membrane selects the particles, the type of particles that it allows, while it never allows another types of particles, for never ever. So that's why it is known as selective. What is semi-permeable? That means these type of particles that it allowed to pass. In sometimes it allows for that type to enter, in another time it stops allowing it, blocks it from entering to the cell or from passing through the plasma membrane to exit outside the cell. So the semi-permeable, it means the type of particles that it usually allows, sometimes it stops allowing it to enter or to exit the cell. While selective, that means it allows to a type and never allows to another type. بالنسبة إلى النوع الثالث وهو الأغشية ذات النفاذية الاختيارية، يعني هنا تسمح بعبور المواد اختياريا تبعا لحجم جزيئاتها، ومثال عليه أيضا الغشاء البلازمي. وأيضا نحن نلاحظ أنه الغشاء البلازمي حتى في تعريفه، حتى في شرحه ضمن صفحة 10 يذكر بأنه selectively permeable، يذكر أنه اختياري النفوذية. هل صحيح أنه الغشاء البلازمي هو اختياري وهو شبه نفاذ؟ وكيف؟ الغشاء البلازمي هو اختيار النفوذي وهو شبه نفاذ في آن واحد. كيف ذلك؟ وما الفرق؟ أولا هو اختيار النفوذية فهو يسمح لمواد بالمرور ولا يسمح لأخرى أبدا، كما في جزيئات البروتينات. جزيئات البروتين تكون عملاقة كبيرة الحجم، فيصعب مرورها بعملية النفوذية بالpermeability، يصعب مرورها عبر الغشاء البلازمي، فلا يسمح لها بالنفوذية نهائيا. بينما يسمح لمواد أخرى كالسكر وغيرها يسمح لها بالمرور. إذا اختياري أنه سمح لمادة معينة ولم يسمح لمادة أخرى. أما شبه نفاذ مقصود بها المادة التي يسمح لها هي نفسها أوقات يسمح لها وأوقات يمنعها حسب حاجة الخلية. فمثلا إذا كان يسمح للسكر بالمرور فهناك أوقات يسمح لمرور السكر وأوقات يوقف مروره حسب حاجة الخلية، فنقول عليه هنا شبه نفاذ لأنه يسمح في أوقات ولا يسمح في أوقات أخرى. أما اختياري أنه يسمح للسكر نعم ولا يسمح للبروتينات، هذا مقصود بالاختياري وهذا القصد الثاني بالنسبة لشبه نفاذ. So here plasma membrane is semi-permeable and also it is selectively permeable. The fourth example here, or the fourth type of membrane, is the non-permeable membranes. Non-permeable membranes is like nylon, and it stops here where the full stop. That's all. This is this here should be goes down a row. When it says it must be noted, that doesn't belong to the non-permeable membrane. It stops here too, like nylon. Nylon, we know, nylon is an example for non-permeable membranes. Nylon, when we have a package of nylon, when we add water in it, we realize that water never passes through the nylon, so it is non-permeable membrane. إذن المثال الأخير لأنواع الأغشية هو الأغشية التي تكون غير نفاذة مثل غشاء النايلون. نلاحظ أن كيس النايلون عندما نضع فيه الماء فإنه لا ينفذ من خلاله الماء، فهو non-permeable membrane. Here it says, it must be noted that permeability of plasma membrane is affected by what? The first is by internal factors and the second is external factors. Here this note is very important. Sometimes the answer would be as fill in the blanks. The permeability of plasma membrane is affected by a blank here and a blank here. The first blank must be filled with the word internal factor and the second blank with external factors. That means there are factors from inside the cell and other factors from outside the cells. They have effects on permeability. So the permeability of the materials or particles can change by time. Why does it change? Because of some factors. These factors from inside the cell or from outside the cell. For example, I can give an example for these factors, maybe the pH or the temperature, light, and some things like that. The change in the chemical structure inside the cell and outside the cell can affect the permeability of the plasma membrane of that cell. So we have here external factors and internal factors. These two factors can affect the permeability of the plasma membrane. The third type, or the third way for the exchange of materials, here we said the first one was diffusion, the second was permeability, the third one is osmosis. I have an important note here when I want to talk about osmosis and the difference between osmosis and permeability. osmosis is talking about the passage of water, while permeability talks about the passage of particles, ions, and materials. So this is the main difference between osmosis and permeability. إذن لدينا هنا الطريقة الثالثة لمرور المواد عبر الأغشية هي الأوزموزيس أو الأوزموزية أو التناضح. وهناك ملاحظة مهمة ما بين الأوزموزية والنفوذية. الأوزموزية هي تتحدث عن مرور الماء عبر الأغشية، أما النفوذية تتحدث عن مرور المواد أو الجزيئات أو الأيونات عبر الأغشية. فإذا عندما نتحدث عن مرور الماء نقول الأوزموزية أو التناضح، وعندما نتحدث عن مرور المادة عبر الغشاء نقول نفوذية هذه المادة. Osmosis is defined as movement of water particles through semi-permeable membranes. Semi-permeable membranes like plasma membrane. So it is defined as the movement here of what? Of water particles through semi-permeable membranes like plasma membrane. According to what? According to variance in concentration. إذن هي حركة جزيئات الماء خلال غشاء اختيار النفوذية، مثال عليه الغشاء البلازمي أو كما قلنا شبه نفاذ هنا كالغشاء البلازمي، تبعا لأي شيء؟ تبعا لاختلاف التركيز. وتتم حركة جزيئات الماء هنا وفق قوانين الانتشار أو وفق قانون الانتشار. So the movement of water particles is done according to what? To diffusion law. إذا تتم هنا عملية التناضح أو حركة الماء هنا وفقا لقانون الانتشار. Because osmosis is a case of diffusion. إذا لماذا يقول هكذا؟ لأن التناضح هي حالة بالفعل، هي حالة من حالة الانتشار. When do we say that diffusion occurred here? When the particles are moving from high concentration to low concentration. That means these particles, if they were water or materials, are moving depending on diffusion process or diffusion case. إذا هنا نجد أن التناضح هي حركة جزيئات الماء عبر الأغشية، وتحصل هنا اعتمادا أو تبعا لاختلاف التركيز ومعتمدة على قوانين الانتشار، لأن هي حالة من حالات الانتشار بالفعل، لأن تنتقل هنا المادة من أماكن ذات التركيز العالي لها إلى أماكن ذات التركيز الواطئ لها. Here we have an experiment also for osmosis, the osmosis mechanism. To clearly illustrate osmosis, we can do a simple experiment using semi-permeable membranes, like cellophane. Attached at the end of a funnel. Here I have an important note. First, when he mentioned semi-permeable, it gave the example plasma membrane. Here, when he mentioned the semi-permeable, the example was cellophane. So the student may ask in the exam, which is the right answer if I needed to put an example for semi-permeable membrane? The answer for semi-permeable membrane here could be plasma membrane. Also, we can mention the cellophane. Cellophane is also a correct answer for semi-permeable membranes. Also, I have another important note here. Another important note. If the question was explain why a plasma membrane is considered a semi-permeable membrane, the answer would be because these membranes do not allow equal passage of solutes as level of solvents. That is the answer. if another explain was, why is plasma membranes considered as selectively permeable membranes? The answer would be because this membrane allows selective passage of materials according to size of their particles. And now we return to osmosis. As we said, cellophane is an example for a semi-permeable membrane. Maybe the student asks about the cellophane. Is it the same cellophane that we use in the kitchen? The answer is no. Because the cellophane that we use in the kitchen is composed of aluminium, and it is a type of non-permeable membrane. Cellophane here is a specific type of tissue. It is a specific type of tissue. It is manufactured especially for laboratories for doing these types of experiments. It is a tissue made in a way that makes it really semi-permeable, so we can use it for experiments in laboratories and to explain how osmosis mechanism occurs. So it is a specific type of tissue. It is not the cellophane that we use in the kitchen. It's a different type, but here at this moment I don't have this type of membrane, but I have to give this a note so you can have the imagine what type of tissue this is, the cellophane that is used in this experiment. So for the osmosis mechanism, here we're going to use a semi-permeable membrane. This semi-permeable membrane is cellophane. The cellophane, we attach it to the end of a funnel. A funnel is a glass funnel. Here we have a funnel. This funnel, it is a glass funnel, and we bring the cellophane here and we attach it to the bottom here of the funnel, and we secure it so it doesn't fall. So the cellophane, which is a semi-permeable membrane, is attached at the end of the funnel. The funnel is filled with distilled water. Here is the distilled water inside the funnel. Here it confirms that it is distilled water. That means it does not have salt ions or sugar ions or something like that. So that's why we use distilled water. Then this funnel is placed in a glass basin. Here we have a basin. Maybe I will make it more shorter. Here, a glass. And the water is here. This basin here is a glass basin. This is the funnel, which has inside it distilled water. Water level should be the same in the basin and in the funnel. Here, the water in the basin and in the funnel are at the same level. Also, here the glass basin has also distilled water. That means the concentration of water outside the funnel and inside the funnel are equal, because both of them does not have any types of sugar or salt ions or something like that. Water level should be at the same as it said here. It's the same. That means they are equal inside the basin and the funnel. Now we are going to bring a beaker that has inside it distilled water, and we add to it sugar. So this will be a solution. It will be a sugar solution. This sugar solution is going to be added to this funnel. When we add the sugar solution here, we will realize that the water here from the basin starts to enter to the funnel passing through this semi-permeable membrane, which is the cellophane. It says here water level, it indicates that water passes cellophane membrane to the sugar solution in the funnel, causing hydrostatic pressure. I'm going to explain that here. Water here, after adding sugar solution to the funnel, the water, which is the distilled water in the basin, is going to enter through the cellophane into the funnel, passes through the cellophane. When water enters here, and as we said, osmosis is the movement of water particles, so here is going to create a pressure. The pressure from the basin towards the cellophane. This pressure here from the water and upwards is osmotic pressure, because we said the movement of water. So osmotic pressure from the basin into the funnel. Water continues entering, so the level of water is not going to be equal now. Inside the basin is going to be lower, while inside the funnel is going up and up. The water that enters the funnel is going to make a pressure from inside the funnel towards the cellophane, from up to down. This pressure here is hydrostatic pressure. So now we have two pressures. The first one is the osmotic pressure from the basin into the funnel, and the other is hydrostatic pressure from inside the funnel towards the permeable membrane. When sugar solution here, it said it is added and the water starts to enter, afterwards hydrostatic pressure. This is the hydrostatic pressure here. Water particles is going to stop. When does that happen? When hydrostatic pressure becomes equal to osmotic pressure. Then here, when are water particles going to stop from entering the funnel? The answer would be when hydrostatic pressure becomes equal to osmotic pressure. So when these two pressures are equal to each of them, the water stops entering the funnel. Solutions can be classified according to osmotic concentration into 3 types. إذن هنا عندما قمنا بإجراء تجربة بسيطة باستخدام غشاء ذو نفاذية اختيارية أو شبه نفاذ الذي هو غشاء السلوفان، تم ربط هذا الغشاء بإحكام عند الجزء النهائي لهذه القمع الزجاجي، وتم ربطه بإحكام، ثم تم ملء هذا القمع بالماء المقطر ووضع في حوض زجاجي يحتوي على أيضا ماء مقطر، بحيث أصبح مستوى الماء داخل القمع وخارجه بشكل متساوي. عندما تم إضافة محلول سكري إلى هذا القمع، تم ملاحظة ارتفاع مستوى الماء داخل هذا القمع في أنبوبة القمع الزجاجي، فهذا يشير إلى أن الماء هنا بدأ يمر خلال غشاء السلوفان إلى المحلول السكري في داخل القمع، فتسبب بالنتيجة هنا ضغط. دخول الماء من الإناء إلى القمع ولد ضغط أوزموزي. كما قلنا الأوزموزي هي حركة الماء عبر الأغشية اللي هي سيمي بيرميبل. إذا تولد هنا ضغط أوزموزي، لكن الثقل هنا الذي أصبح في داخل الفنل داخل القمع ولد ضغطا آخر معاكس اللي هو الضغط الهيدروستاتيكي. تولد هذا الضغط الهيدروستاتيكي من داخل القمع باتجاه غلاف قطاع السلوفان. إذن ثم في لحظة ما سيتوقف دخول الماء من الإناء الزجاجي إلى القمع. متى هذه اللحظة التي يتوقف فيها الماء؟ هذه اللحظة هي عندما يتساوى الضغطان، الضغط الهيدروستاتيكي والضغط الأوزموزي. وهذا سؤال جدا مهم وقد يأتي بهيئة تعليل explain when are water particles going to stop from entering the funnel. طبعا هاي متى؟ في تجربة تناضح، فنقول عند تساوي الضغطان الهيدروستاتيكي والأوزموزي. إذن بعد ذلك أيضا هنا عندما يطلب التجربة يجب أن نرسم معها الرسم حتى لو لم يطلب الرسم في السؤال. Also I should mention that if in the exam it asks about to illustrate the osmosis mechanism, the student should draw the osmosis mechanism, which is this figure here, even if it was not asked for it, but the student must draw it because there is no experiment without a drawing. Now we come to solutions and the types of solutions. So solutions can be classified according to what? Solutions can be classified according to osmotic concentration into 3 types, and each of these types has a particular effect in the cell. إذا تقسم المحاليل تبعا لتركيزها التناضحي إلى 3 أنواع، ولكل منها تأثير خاص في الخلية. So what are these 3 types of solutions? This is part of the osmosis. These informations here is part of osmosis. So now we are going to take the types of solutions. Here the first type is isotonic solution. The second type here is hypotonic solution. The third type is hypertonic solution. So what is isotonic solution? Isotonic solution, whereby concentration of water outside the cell is equal to concentration of animal cell cytoplasm. The cell neither loses nor gains water. إذن هنا لدينا المحلول متعادل التركيز. متعادل, what does it mean? Iso. Here when we have a cell, so it was here I'm drawing an animal cell here, surrounded by a plasma membrane. When the concentration of water inside the cell and the concentration of water outside the cell are equal, so we say that this cell is inside an isotonic solution. Usually here the cell never loses or gains water. Really, the fact here is the cell loses and gains, but it loses and gains equal quantities. So it looks like it never gains or loses, but really it gains and loses. But in the exam, the student should write what is here considered in its book. So you can't say that water enters, you say that the cell neither loses nor gains. But I wanted to say the real fact here is it really loses and it gains, but in equal quantities, so we never realize it. It needs to be more inside. When we are looking at it from the microscope, we should concentrate during looking for water particles, if they are really entering or not. But the real fact, they are entering, and it enters and it also loses water, but in same quantities that we cannot really realize it easily. إذن هنا في هذا المحلول متعادل التركيز، فيه يكون تركيز الماء خارج الخلية مساو لتركيز الماء في داخل سيتوبلازم الخلية. فالخلية عادة لا تكسب ولا تفقد، لكن بالحقيقة الخلية تكسب وتفقد لكن بكميات متساوية وبحركة سريعة نكاد لا نراها، لكن هي بالحقيقة هي هناك حركة لكنها سريعة جدا. لكن يلتزم الطالب بمعلومات الكتاب، وهي أنه يقول أنه الخلية لا تكسب ولا تفقد مثل ما مذكور في المنهج. This is the isotonic solution. Before we go to the hypotonic solution, I need to explain something important. In the exam, when the question is about animal cells and plant cells that are put in isotonic solution, so we must draw this drawing here in figure 1.18. It talks about osmosis in plant and animal cells. Here I'm going to draw it, so it will be clearly for you. Here is an animal cell. When an animal cell, this is the nucleus here, plasma membrane, and this is the cytoplasm. An animal cell here, animal, when it is put in isotonic solution, we realize that we are here in this image, this arrow enters the cytoplasm, and other arrow also enters, while the third one exits and the fourth one also exits. We can realize that here. This one and this one, both of them enter to the cytoplasm, while these two arrows exit from the cell. That means it really loses and gains, but in equal concentrations. This is an animal cell when it is put in isotonic solution. Now I need to draw a plant cell when it is put in isotonic solution, so it looks natural and normal. Here is a plant cell. This is the plasma membrane. It is surrounded by a cell wall. This is the cell wall. The cell has here a nucleus and a big or a huge vacuole that has a cell sap. This is the vacuole, central vacuole, and here is the cytoplasm here. This is the cytoplasm. Now, when I'm going to draw the arrows, the same as in the animal cell, this is a plant cell here. We were drawing the plant cell. The arrows here, it enters to reach the vacuole. The second one also enters here, and two exits. So that means when I'm drawing a cell in isotonic solution, I should clarify it very well by drawing two arrows entering the cell and two arrows exiting from it, also in the plant cell. Maybe in the exam it says draw a muscle cell, a red blood cell, white blood cell, any type of cell that is animal cell. All of them are animal cells that I mentioned now. And a red blood cell that has been put in isotonic solution. It's okay. Red blood cells are animal cells. That means he wants me to draw the animal cell. And that's all. And when it asks about a plant cell, so I must draw the plant cell. If in the exam the question was a cell was put, but he never mentions if it was an animal or a plant cell, that means I must draw both of them. But if the question was asking about only plant cells, so the drawing must be only about the plant. If it was about animal cells, so the drawing must be only about the animal cell, without drawing the plant cell. Now, when we are talking about the second type of solutions, which is the hypotonic solution. Hypotonic solution, this solution has low concentration of non-permeable solutes compared to solutes in cytoplasm. The cell gains water. This leads to distension of the cell and then rupture, especially in animal cell. الهايبوتونيك solution هذا المحلول بيش يتميز؟ يحتوي هنا على تركيز واطئ منخفض من المواد الذائبة التي تكون غير نفاذة. إذا ما تم مقارنة هذه المواد بالمواد الذائبة في سيتوبلازم الخلية، يعني القصد هنا بالهايبوتونيك سوليوشن إنه الخلية وضعت في محلول، خلية وضعت في محلول هنا. المحلول يحتوي على تركيز قليل من المواد، بمعنى تركيز الماء هنا عالي. هذه الخلية وضعت في إناء يعني في محلول، والمحلول يحتوي على تركيز قليل من المواد غير الذائبة، يعني يحتوي على كمية ماء عالية، مقارنة مع الخلية نفسها اللي هي تحتوي بالمقارنة مع الخارج هنا تحتوي على ماء أقل ومواد غذائية أعلى. إذا هنا كانت المادة الغذائية قليلة، إذا المادة الغذائية في الداخل عالي. إذا كان الماء هنا عالي، يعني الماء في الداخل قليل. عندما يتحدث عن hypotonic solution، إذن هو المحلول نقول عنه واطئ التركيز. واطئ التركيز بأي شيء بالمادة، بمعنى أنه من ناحية الماء هو عالي بالماء، هو واطئ بالمادة، إذن هو عالي بماذا؟ بالماء. بما أن موضوعنا هو الأوزموزس الذي يتحدث عن انتقال الماء، إذن سوف نتابع أين يكون الماء عالي. سوف يتجه بالأماكن التي يكون فيها الماء قليل. المحلول واطئ التركيز بالمادة، لكن من ناحية الماء هو عالي بالماء. موضوعنا هو الأوزموزس، إذن نلاحظ انتقال الماء، وقلنا انتقال الماء بالأوزموزس من أماكن ذات التركيز العالي بالماء إلى أماكن ذات التركيز الواطئ بالماء. إذن هنا سوف ننتبه إلى كيف يتجه الماء من أماكن ذات التركيز العالي به إلى أماكن ذات التركيز الواطئ به هو أيضا الماء. Here in hypotonic solution, hypotonic, that means the solution here has a low concentration. Hypo is low concentration of materials, but it has a high concentration of water. So because it has high concentration of water and our subject is osmosis, that means it's going to realize water particles, and usually water particles in osmosis mechanism, they move from high concentrations of water to low concentrations of water. And we said that here we put this cell inside a hypotonic solution which has low concentration of materials but high concentration of water. So water is going to move from high concentration to it towards the low concentration of water. That means the cell here is going to gain water. When it's going to gain water, that means it is going to explode if water continues entering to it without stopping. إذن هنا بما أنه وضعت الخلية في محلول واطئ التركيز بالمادة لكنه عالي التركيز بالماء، فالماء سوف ينتقل من خارج الخلية من هذا المحلول إلى داخل الخلية، فيؤدي إلى قد يؤدي إلى انفجارها. أكيد الخلية سوف تنتفخ وقد يؤدي إلى انفجارها إذا لم يتوقف دخول الماء إلى داخل الخلية. Now we are going to draw a cell that is put in hypotonic solution. In hypotonic solution, the cell is going to explode like this. Water enters, continues to enter, afterwards it explodes like that. Now this cell, this is the nucleus of the cell, and this is the plasma membrane, and now the arrows shows what happens. How does it explode? Because water enters 1, 2, 3. So we draw 3 arrows entering and 1 arrow exiting. So what happens here is the extension of the cell and rupture. This is for the animal cell. What happens to the plant cell here, cell wall? The plasma membrane here, the nucleus, and the central vacuole, also this cytoplasm here. Now we're going to draw the arrows 1, 2, 3, while one of these 4 arrows is directed from inside to outside. Its direction here, like that. The plant cell didn't explode. Why? The plant cell here doesn't explode because it is surrounded by a cell wall, and we know cell wall is as we said here, it is permeable membrane, so it allows water and particles to to pass through it. So that's why it may make these materials or particles pass easily, so it does not rupture. So here we see the cell wall, the plasma membrane, and these arrows, 3 of them are entering and 1 of them is exiting. Also for the animal cell, 3 of them are entering and 1 of them is exiting. In the exam, when it asks about an animal cell or plant cell put in hypotonic solution, it wants you to draw it. So normally we draw the normal state first when they are put in isotonic solution. Then we draw the hypotonic solution, the cell put in hypotonic solution, so we can clarify the change that has happened between the normal features of a normal cell and the changes that have happened in hypotonic solution. And now we come to the last type of solution, which is the hypertonic solution. Hypertonic solution, this solution has high concentration of solute materials compared to cytoplasm. المحلول عالي التركيز، هذا المحلول يتميز بتركيز عالي من المواد الذائبة بالمقارنة مع السيتوبلازم. So here we have a cell put in hypertonic. Hyper. That means it has high concentration of materials outside, high concentration, which means that water is in low concentration. If the materials here are high, when we say hypertonic, that means these materials inside the cell are low. If water here is low, that means water inside it is high. And as we said before, our subject is osmosis. So when we are talking about hypertonic solution, water here is high while water here is low, so water is going to move from the inside to the outside. So what happens to the cell? It will shrink. Hypertonic solution here, it says, thus, direction of water is from cytoplasm to the outer solution. This causes shrinkage of the cell. The size of plant cell compared to animal cell does not change when placed in hypotonic solution. Why? Because plant cells have cell walls. Here, when we see the image inside the book, This is an animal cell. Shrinkage is clarified here, while plant cell we cannot realize the shrinkage, but it's type of shrinkage that has happened inside it only, not from the outside. Why? Because it is surrounded by a cell wall. What really happens here is this cell will go through shrinkage like that. This is the plasma membrane, the nucleus, and why shrinkage has happened after we put it in hypertonic solution. The water, 1, 2, 3, only 1 enters, 3 exits, 1 enter, because the water inside the cell is in high concentration compared with the water outside the cell. So the cell loses the water. So water exits the cell and starts to shrinkage. While for a plant cell here, if we put a plant cell in a hypertonic solution, this is the cell wall, never changes. Here is the plasma membrane. See how it pulls itself away from the cell wall? So shrinkage happened here inside, but we cannot realize it easily because it is surrounded by cell wall. And here is the vacuole and the nucleus. Also, we are going to draw the arrows, which are very important. 1, 2, 3 exit, and only 1 enters. Here is a plant cell that has been put in hypertonic solution as well. In the exam, if it asks about drawing a plant or animal cell put in hypertonic solution, at the first we should draw the state of isotonic solution, then we draw the hypertonic to show the changes that have happened to a normal cell into a cell that has been put in hypertonic solution. What happened here in plant cell is called plasmolysis, when the plasma membrane goes away from the cell wall. So, what is plasmolysis? Plasmolysis is a process where cell membrane moves toward the center of the cell. That results from water leaving the cell, which causes shrinkage of the cell placed in hypertonic solution. That occurs in plant cells. If the question was explain the size of plant cell compared to animal cell does not change when placed in hypertonic solution, the answer would be because plant cells have cell walls that doesn't get affected when plant cell is placed in hypertonic solution, while cell membrane merely moves away from the cell wall. And a question here, can a plant cell that has gone through plasmolysis, can we change its state and return it back to normal? Can we get rid of plasmolysis? The answer is yes. What happened here in plant cell, as we said, it is plasmolysis. The other state that is releasing it from plasmolysis is deplasmolysis. Deplasmolysis. What is deplasmolysis and when does it occur? A plant cell that has gone through plasmolysis, when we take it and put it in water, or we put it in isotonic solution, hypertonic solution, it will gains water again. So we are going to release the plasmolysis, and it returns back to a normal state. So deplasmolysis, it is a reversed process to plasmolysis, when the cell goes back to its previous state after adding solution. This is deplasmolysis. I repeat it. Define deplasmolysis. It is a reversed process to plasmolysis, when the plant cell goes back to its previous state after adding solution. So here it says plasmolysis, when the cell membrane merely moves away from the cell wall. It results from water leaving the cell, but when the solution is added to the cell, it goes back to its previous state. This reversed process is called deplasmolysis. Now we have completed 3 ways of exchange of materials. The first one was diffusion, the second was permeability, the third was osmosis. Now we come to active transport, which is the 4th way of exchanging materials. D is active transport. إذن أعيد باللغة العربية. عندما تم وضع الخلية النباتية والخلية الحيوانية في محلول عالي التركيز، لاحظنا أن عندما يقال محلول عالي التركيز معناه أنه وضعناها في ماء أو في محلول يحتوي على تركيز عالي من المادة لكنه تركيز واطي من الماء. بما أن تركيز الماء في داخل الخلية أعلى مما في الخارج، فيميل الماء إلى الخروج من الداخل إلى الخارج حسب قانون الانتشار بعملية التناضح، فيؤدي إلى انكماش الخلية، وقد يؤدي أيضا إلى موت الخلية إذا استمر الانكماش. بالنسبة للخلية النباتية لا يمكن أن نراها بالصيغة الواضح هذا الانكماش، بسبب أنه محاطة الخلية النباتية بالجدار الخلوي، لكن بالحقيقة أنه تم انسحاب الغشاء البلازمي عن الجدار الخلوي، فهذا الانكماش واضح إذا أردنا أن نراه تحت المجهر. لكن هذه العملية التي تحصل في الخلية النباتية تسمى بالبلزمة عند انسحاب الغشاء البلازمي عن الجدار الخلوي. لكن هل يمكن إعادة الخلية إلى الحالة المعاكسة؟ نعم ممكن، وتسمى الحالة المعاكسة بإزالة البلزمة. وإزالة البلزمة تحصل عندما نقوم بوضع هاي الخلية التي حصل فيها بلزمة، نضعها في محلول أو في الماء أو في محلول متعادل التركيز أو في محلول واطي التركيز، فإنها تبدأ بسحب الماء من جديد، فيدخل الماء إلى الخلية، فيعود الغشاء البلازمي إلى وضعه الطبيعي، فيمكن إزالة البلزمة بعد أن يتم وضع الخلية في محلول واطي التركيز أو في الماء. العملية الأخرى لطرق انتقال المواد عبر الخلية هي النقل الفعال، النقل النشط أو النقل الفعال نسميه. When we reached to active transport, I only want to mention an important thing, that the 3 ways that we have mentioned, the diffusion, the permeability, the osmosis, all these ways does not need ATP. That means it does not need energy for doing these mechanisms or processes, while active transport needs energy. So that means it needs ATP to take from the cell, because what is going to pass is reversible to the diffusion law. In active transport, here cells sometimes absorb materials from the outside environment, although the concentration of these materials inside the cells is higher than outside. Why? To perform. Why that happens? Really, active transport occurs when the cell has some needs. Its needs may be ions, particles, molecules, so it has some needs, and these needs are maybe they are outside the cell, or sometimes they are inside the cell, and they need to move from low concentration to high concentration. And usually if these materials are in low concentrations, they do not move towards high concentration. So how can a cell do that? So the cell does that by using energy, ATP, and it needs something to help it with it, and what aids in that is the carriers that it has in the plasma membrane. إذا هنا نجد أن الخلايا تمتص أحيانا بعض المواد من محيطها الخارجي، بالرغم من أن تراكيز هذه المواد في تلك الخلية في داخلها هي أعلى مما في الخارج. ولكي تقوم هذه الخلية بهذه الحالة تقوم بها عادة بسبب حاجة الخلية لهذه المواد. نحن نعلم أن إذا كانت المادة في داخل الخلية مثلا بتركيز عالي وفي الخارج هي واطئة، فأحيانا تدخل الخلية مواد نفس هذه المادة أيضا من الخارج نحو الداخل، رغم أنه هي عالية في الداخل، لكن بما أنه عكس قوانين انتشار هذه العملية فلا يمكن أن تتم دون صرف طاقة، فتحتاج إلى ATP. لكن نفس الوقت الخلية لا تقوم بذلك إلا إذا كانت لها حاجة بذلك، تحتاج إلى رفع معدلات أو تراكيز هذه المادة حتى تقوم بوظائفها بشكل أفضل، فتقوم بسحب كميات إلى الداخل رغم أنه هي أصلا هي هذه المادة عالية في داخلها، لكن تحتاجها الخلية لأجل إنجاز وظائفها بشكل الأمثل. So here we see that the cell sometimes absorb materials, as we said, even if these materials are with high concentrations inside the cell. And maybe an example here we have is the thyroid gland. We have a thyroid gland, and that is responsible for metabolism that occurs inside our cells, our organs, the cells of our organs. So it needs high concentrations of iodide to create thyroid hormones. So the thyroid here, the iodide is in high concentrations inside it, inside the thyroid gland, but whenever it realizes that there are iodide in the blood, it's going to absorb more iodide, because it needs iodide in high concentrations inside it till that time it can start making thyroid hormones, because low levels of iodide inside it will make low levels of thyroid hormones, which is not perfect for the thyroid gland. لدينا مثال على ما يحصل في النقل النشط أو النقل الفعال كما في الغدة الدرقية لدى الكائن الحي. الغدة الدرقية تحتاج إلى اليود، أيونات اليود تحتاجها بكميات عالية بتراكيز عالية، فحتى لو كانت بتراكيز عالية داخل الغدة لكن تحتاج إلى كمية أكبر حتى تؤدي وظيفتها بكفاءة عالية. أحيانا تستهلك جزء من عنده، لكن يبقى بالداخل هو أعلى مما في الخارج الذي هو في الدم يعني، لكن تحتاج خلايا الغدة إلى كميات بنسب عالية جدا حتى تستطيع صناعة هرمون الثايرويد الذي يسيطر على وظائف الأعضاء المختلفة، خلايا أعضاء الجسم المختلفة. and إذن أيضا هنا يؤكد إنه الخلايا، طبعا ممكن يكون هنا التعليل لدينا تعليل أو يأتي في الامتحان يقول تمتص الخلايا أحيانا بعض المواد من محيطها الخارجي بالرغم من أن تراكيز تلك المواد داخل الخلية أعلى منها في الخارج. الجواب يكون أولا بسبب حاجة الخلية لذلك، وثانيا لتوفر الكاريرز اللي هي هذه الحوامل اللي هي البروتينات، اللي هي البروتينات الحاملة التي تساعد في عملية النقل الفعال. فوجود هذه المواد الحاملة في الغشاء البلازمي هي التي تسهل، لكن الأمر الأول هو سببه هو حاجة الخلية، والثاني هو لوجود ما يساعدها في ذلك وهو المادة الحاملة الكارير الموجودة في الغشاء البلازمي. So here the question will be, cells usually absorb some materials from the outside environment in spite of the concentration of these materials inside the cell is higher than the outside. The answer would be according to the cell needs, as it has carriers in the cell membrane, can move inside and outside the cell to integrate with needed materials, particles or ions. This requires energy, which is supplied by ATP. So usually here the cell takes ATP and uses the carriers to move these it moves these carriers by using the ATP to allow the entering of materials from low concentration to high concentration. And here it says that these carriers moves to the inner surface of the membrane. The carried materials separate in cytoplasm. This process requires energy, which is supplied by ATP. Usually here for the cell, here we're going to draw plasma membrane. These are phospholipids of the plasma membrane that are separated by protein particles, which are known as carriers. So that means in the exam when it asks about the chemical structure for the carriers, the answer would be protein particles or protein. That's all. Here is the other carrier. In this image here, it shows the inside of cell, while here is the outside of cell. Here they are the ions or particles which are outside they are in high concentration. Inside the cell they are in low concentration. These particles here enter to to integrate with the protein particles. That means it integrates like that with it. As it enters like that, integrates with the protein particles, then the protein particles changes its direction like that. So when it changes its direction, it will separate from the protein particle, and it shows it here, this arrow here. It shows how this particle that has entered, it's going to go outside, but these protein carriers, they needed what? They needed ATP to take from the cell. Because why does it need ATP? Because it is reversed to the diffusion law. That means it's going to take these particles from low concentration to high concentration. Sometimes students ask, is this here, is it right? Is it right that active transport occurs from inside to the outside? The answer is yes. Active transport occurs from inside to outside or from outside to inside. It is known as active because it needs to use ATP to exit materials or to enter materials, reverse to diffusion law. So this is an appropriate without anything. There isn't anything wrong with it, and this is an example of active diffusion on a cell membrane, if it was from the inside or from outside to inside. While this image here that talks about passive diffusion is reversed to active. Passive, it was what usually happens in plasma membrane. What happens depending on diffusion law. That means it doesn't spend ATP here, while in active diffusion or active transport it needs ATP because it is reversed to diffusion law. Passive diffusion, here we can realize that the particles are in high concentration. This arrow here shows how the particle enters to the inside the cell from high to low. So here it does not spend energy and it depends on diffusion law, while here it's reversed to diffusion law and needs ATP. إذن في عملية النقل الفعال هنا نجد الغشاء البلازمي وتفصله هنا الشحوم المفسفرة تفصل الجزيئات البروتينية، والجزيئات هنا عندما تنتقل من أماكن ذات التركيز الواطئ لها إلى أماكن ذات التركيز العالي لها هنا ترتبط مع المواد الحاملة التي تركيبها الكيميائي هي بروتين، ترتبط معها ثم يتغير اتجاه البروتينات الحاملة إلى الجهة الثانية وبصرف طاقة حتى تنقله هذه الجزيئات من أماكن ذات التركيز الواطئ لها إلى أماكن ذات التركيز العالي لها، سواء كانت من داخل الخلية إلى الخارج أو من خارج الخلية إلى الداخل، نفس الشيء، كلاهما يعتبر نقل نشط ما دام هو عكس قوانين انتشار ويتم فيه صرف طاقة. أما بالنسبة هذه العملية اللي هي الباسيف ديفيوجن، الانتشار الميسر، هذه اللي هي الطريقة العادية والطبيعية التي يقوم بها الغشاء البلازمي بنقل المواد من أماكن ذات التركيز العالي لها إلى الواطئ التي هي ضمن قوانين الانتشار ولا تحتاج إلى صرف طاقة. So this was the active transport, which is the fourth way of exchange of materials. If the question was, mention the importance of the carriers, the answer would be carriers integrate with needed materials, particles or ions, and move inside and outside the cell according to the cell needs. Another question, what is the location of carriers? The answer would be in the cell membrane between phospholipids. Another question, what is the chemical structure of the carriers? The answer would be proteins. Another question is, cells that perform the function of active transport are characterized by the fact that they contain extensive mitochondria. The answer would be because active transport process requires energy, which is ATP. This energy is provided by mitochondria. The fifth type or fifth way of exchanging materials is phagocytosis. Phagocytosis. Phagocytosis means that the cell is eating. In a previous lecture, we mentioned the lysosomes and how they have a role in phagocytosis by digesting what happens in the digestion in the cell. Here, so phagocytosis is cellular eating. It is a common way of eating nutrition among protista like amoeba. It is also used by white blood cells. So examples for phagocytosis, what occurs in protista. An example for protista is amoeba. And the second is in another type of eukaryotic cells inside a human being is the white blood cells. Two examples here for phagocytosis process. cells here devouring remaining cells and germs in the blood. This process is done when cell membrane is pouch-like that surrounds the solid matter. Then this pouch separates from cell surface and moves inside cytoplasm. The contents are digested by the enzymes secreted by lysosomes in the cytoplasm. That means if here we have this is a plasma membrane, and here we have some solid particles, and this is the plasma membrane. When these solid particles are found in the environment outside the cell, the cell membrane makes this pouch-like. It will surround this solid matter. Then this pouch will separate, and this is the solid matter inside it. So the pouch will separate from the cell surface and moves inside this cytoplasm. This formed here is known as food vacuole. Then a lysosome here, which has about more than 40 types of enzymes, will come near this food vacuole and attaches to it and release these enzymes on this food solid particles and digest it. As it says here, the contents are digested by the enzymes secreted by lysosomes in the cytoplasm. So here it needs lysosomes to digest the food that has been surrounded by the cell membrane and forming the food vacuole. So the food, when the question is the solid particles inside the food vacuoles, how can they be digested? The answer would be depending on the enzymes that have been released from the lysosomes. And if the question was illustrate how the mechanism, phagocytosis mechanism occurs, we can illustrate it by this process is done this is the first one. This process is done when cell membrane is pouch-like that surrounds the solid matter. Second, this pouch separates from cell surface and moves inside the cytoplasm. The fourth is the content are digested by enzymes secreted by lysosomes in the cytoplasm. Now we come to the sixth way of exchanging materials, which is pinocytosis. The difference between pinocytosis and phagocytosis. Pinocytosis, it is similar to phagocytosis, but here we are depending on liquid material. Here, when this cell is found in an environment that has a liquid material, so the plasma membrane also is going to surround this liquid material. This is liquid material. while here are solid matter or solid particles. This is the plasma membrane. The plasma membrane surrounds this liquid material by making an invagination. Here it made a pouch-like. Here happens it makes a small invagination. This hole surrounds the liquid matter and becomes inside the cytoplasm of the cell. This is known as pinocytic vesicle. While here food vacuole was formed, here is a pinocytic vesicle is formed. Here is a vacuole, here is a vesicle. The difference between the two of them. The cell membrane surrounded the liquid material and formed a pinocytic vesicle. Here we don't need lysosomes, while in phagocytosis we need lysosomes. الطريقة الخامسة كانت البلعمة أو الأكل الخلوي، والطريقة السادسة كانت الشرب الخلوي. الأكل الخلوي كما وضحنا يحصل في الطليعيات كما في الأميبا، ويحصل أيضا في كرات الدم البيض. هذه أمثلة على كائنات يحصل بها فاغوسايتوسيس. وهنا لدينا الغشاء الخلوي، وموجودة هذه الخلية ضمن محيط يحتوي على مواد صلبة، فيقوم الغشاء الخلوي بإحاطة هذه المادة الصلبة، ثم هنا يعمل جيب معين في الغشاء البلازمي ويحيط بالمادة الصلبة، ثم تنفصل عن الغشاء البلازمي وتدخل إلى السيتوبلازم بهيئة فجوة غذائية. أما بالنسبة للمادة السائلة هنا أيضا تحاط بالغشاء البلازمي، لكن يعمل جيب ويحيط بالمادة السائلة وينفصل مكونا حوصلة. هنا يكون فجوة غذائية، هنا يكون حوصلة، كلاهما يدخلان في السيتوبلازم. بالنسبة للفاغوسايتوسيس نحتاج إلى اللايسوسوم. الإنزيمات الحالة لللايسوسوم هي التي تقوم بتحليل المواد الصلبة. أما بالنسبة للبينوسايتوسيس فلا نحتاج إلى وجود اللايسوسوم لإتمام العملية. هذا هو الاختلاف ما بين البلعمة والشرب الخلوي. فالبلعمة تحصل للمواد الغذائية الصلبة، والشرب الخلوي يحصل للمواد السائلة. وأيضا من ضمن الملاحظات أن الفاغوسايتوسيس هي وسيلة لإدخال البروتينات والتي هي الجزيئات الكبيرة التي قلنا لا يمكن لها أن تدخل بعملية النفوذية، لا يمكن لها أن تمر بعملية النفوذية. The last way of exchanging of materials is exocytosis. As we said, materials enter the cell by phagocytosis, but when they exit from the cell, they exit by exocytosis. This term is used to describe the releasing of some materials outside the cell. This process takes place in various cells. Why? To get rid of undigested leftovers from phagocytosis, or to secrete some hormones. The particles that have entered the cell by phagocytosis exit from cells by exocytosis. إذن عملية الإخراج الخلوي هي عملية التخلص أو تحرير بعض المواد من داخل الخلية إلى خارجها، وتحدث هذه العملية في خلايا مختلفة. لماذا؟ من أجل أولا التخلص من بقايا المواد غير المهضومة التي دخلت إلى الخلية بواسطة الإدخال الخلوي اللي هي الفاغوسايتوسيس، أو لإفراز بعض المواد مثل الهرمونات. إذن من يسأل عن أهمية الإكزوسايتوسيس يكون الجواب أولا للتخلص من بقايا المواد غير المهضومة دخلت بواسطة عملية الإدخال الخلوي، وثانيا لإفراز بعض الهرمونات. When the question is what is the importance of exocytosis, the answer would be it takes place in various cells to get rid of undigested leftovers from phagocytosis. The second answer is to secrete some hormones. And now we have took about 7 ways of exchanging materials. As we mentioned before, exchanging of materials was by diffusion, permeability, osmosis, phagocytosis, pinocytosis, and exocytosis. All of these were the exchange of materials which are part of cell activities. The next lecture will be cell metabolism, which is the second type of cell activity. إذن أخذنا 7 طرق لانتقال المواد وعبورها عبر الأغشية، والتي هي إحدى أنواع الأنشطة الخلوية. الدرس القادم إن شاء الله سنأخذ الأيض الخلوي، والذي هو مثال آخر على الأنشطة الخلوية. إذن إلى هنا نأتي إلى نهاية درسنا لهذا اليوم. We have reached the end of our lecture for today, and goodbye for now.